What can I say after all these wonderful talks? So I'm going to try to be very brief with this presentation because the peak is waiting for us. So we have to head up there very soon. So I put together the information or the papers that we have published here during the 11 years that we have been doing this research. And I want to show you very brief what is going on here. So I would like to start with our baseline. So the first time that I put a mask on and jump into the water here, this is the barrens that we saw. This is a barren in La Gomera Island. It's kind of a spectacular landscape just with full of urchins and just bare rock all around. The densities are the maximums are about 12 individuals per square meter, and this phenomenon also it's happening in Madeira, Salvajes, and the Canary Islands. So we have some question here. We start thinking about what are the causes that can lead to such high concentration of sea urchin that barren ground results? And the other question, are they natural phenomena or ones that have only recently begun as a result of changes in the characteristic of marine environment arising from human activity? I think this question have been partially answered today in some areas, but remain valid. This question remain valid, and that's why we are all here today sharing our research. So here we are. We have seen all these cases here with different species that are able to modify the habitat and create these barren grounds all around the world at different ecosystems, from temperate ecosystem to tropical areas. So it's a global phenomenon, and the global warming, I think it's affecting in some ways this phenomenon. In the Canary Islands, what are the consequences of this huge or hyperabundance of urchins? In the Canary Islands, when we have about 4 individuals per square meter, we are sure that the algae not grow. There is a growth of algae, but the algae cover remains always below 30%. So it modifies a key herbivore and modify the habitat, the structure, and the algae. So control very well the algae. Also, the algae richness and diversity go down as the urchins go up. So it's modifying the whole system. Also, as we have previously seen, the macroinvertebrates are also affected, and this is because maybe it's an indirect effect because of the loss of the habitat where these animals used to live. But I think one of the most important things is that when we lost this microalgae system, we lost the service that this system provides to us. Here we have a very important resource that is the old lady, La Vieja, the Sparisoma cretense, and this fish species is affected by the densities of urchin. So as the urchin densities increase, the density of Sparisoma just go down very quick. And also, it modifies the breeding area. So the juveniles of Sparisoma cretense is very low. The abundance of these juveniles is very low at high densities. So it's modifying, the urchins are modifying the service that this macroalgae system provide to us here in the Canary Island. So I would like to start studying or showing you a summary of what we know about the biology of this urchin here. And so understanding the biology of the urchin is very important in terms of to see if the temperature is affecting the population or not. So what regulates urchin population? This is a very difficult question that we have previously seen in the other presentations, and it's a very complex process. So we have a very complex life cycle of the urchin. It's not as complex as a decapod, but it's still complex. And there are larvae that are in the plankton, but then the post-settlement recruits are in the benthic part of the system. And so the life cycle occurs in different habitats, in different ecosystems. So in order to understand how these populations are regulated, we have pre-settlement process that we can summarize. Here, I'm just showing factors that I think are important here in the Canary Island. But of course, there are other factors. For example, the bottom-up modification can modify the larvae supply, the amount of eggs that are produced. The temperature is also important, but physical parameters like the retention areas in these islands are important to drive or to determine the abundance of these urchins. And of course, if we modify the larvae supply, the settlement rate are going to be modified as well, are going to be. And then we have also post-settlement processes that affect the recruitment, like the habitat complexity, predation, wave exposure, storm, disease, rugosity, sedimentation. We have found that all these factors are controlling in some way the abundance of these urchins in the Canaries. But also, these processes vary in a temporal and in a spatial manner. So it adds a little bit of more complexity to the already complex system. So it's very difficult to predict what regulates an urchin population. We have to study many things. So in a way, I just put this slide together to show you in a brief manner the biology of these urchins. So the gonad maturation occurred when there is an increase of algae cover in the benthic system. The spawning occurred during June, July, and August, when there is a decrease in daylight length, and it's coupling as in the Caribbean with waning moon and new moon phases. And the plankton, the larvae are in the plankton during the low abundance planktonic predators and when the temperatures in the water are rising. And the settlement occurs just when the maximum temperatures in the water are reached. So in August, September, and October, we have these huge amounts of settlement, mostly in barrens. So after 3-4 months, we have, after the spawning, we have the recruitment. We can actually found the small animals in the benthic systems. So it's a very, the renewal is very fast of these urchins. So this is the biology. So 3-4 months we can found these very early recruits in the field. So patterns arise from spatial-temporal monitoring. We have been doing monitoring since 2001, even before we have data. And there are very interesting patterns that show up when we study the data that we have. So we can see that the barrens are all around the island. This is a Tenerife, so the purple color means barren. So you can see that the barrens are all around the island. But there is a spatial variation in the urchin abundance, but also in the extension of the barrens. So we have islands like Tenerife Island that have the highest densities of urchins, about 5.5 urchins per square meter. But then there are other islands like El Hierro that barely we have barrens there. There are urchins, of course, but they're not barrens. It's all full of macroalgae, and you're going to have the chance to see these spectacular changes. But also another interesting pattern here is that the densities are lower at high exposure places. So as we have found, and other researchers here in this room have found, that the wave action is very important to limit the action, the grazing activity of this urchin. And an engineer put together this map for me here, and I'm showing the green color mean more wave action, mean more water energy, and the blue areas at the lee part, the lee side of the island mean retention, so water retention. And what we have found, here is a very simple graph, but if you want, I can show you later the results. We found that in these areas, the size and structure of urchins are unimodal, and it's basically skewed to the right. So that means that the renewal of the population is not as frequent as it is in the south part of the island or at this lee part of the island. So there is a physical process here as well, that it's affecting the population of what we are seeing now in the field. So there are barrens here, but they are less dense and also full of all urchins, okay? While in the south, we have younger urchins, and there is a higher renewal of the population. Also, if we go deep, we are going to do these divings, and you're going to be able to see that, for example, in low exposure places, the barrens start around 3 meters depth, while at high exposure places, the barrens start a little bit deeper in around 6, 7 meters depth. And these results coincide with previous results by Fernando Tuya, that he's here today, and he was doing an experiment in a tube with, he changed the wave action, and you can, the model predict this depth very well. So it fits with models as well. If we look at one island like La Palma, the whole island, okay, what we see is this. So the number of hectares occupied by the urchin increase with depth, while the macroalgae beds are just decreasing as the urchins go up. So this is what we found in these islands where the barrens are. This is a typical distribution model. Also, I think that it's involved here that the larvae settlement, and here showing larvae settlement per collector, and at different depth. And we can see very clearly here that the depth level is very important for settles. So there is a higher settlement at deeper waters. Okay, so they settle 10 meters depth, 15 meters depth, but there is almost very few urchins or settles at 5 meters. So the hydrodynamic here is maybe wiping out the larvae from this system. So it's very different from the Mediterranean with the Paracentrotus. Paracentrotus has a different pattern. Here I'm showing PEC a pattern of recruits. So the green are grazing fronts, and the violet are barren grounds. And here I'm showing the complexity variables that were measured to see the complexity of the system. So the adults, the presence of adult, microrugosity, number of holes, the slope, and different algae groups. And what we found here is that the recruits, all the recruits, occur mainly where the adults are. So this is the number means the number of recruits that were counted, and the bubbles mean the density of adults. So they recruit where the adults are. And this have been previously shown by Pierce and Scheibelin that is here, and it's mainly the explanation could be that there are cues in this barren habitat that induce the settlement. But if we take a closer look and we just analyze the data for the barren system, we can see that the recruits appear where there is a higher slope and a higher microrugosity. And you can see there is the opposite way where the adults are. So the adults can provide refuge, but they are not going there because of the adults. They are going there because their habitat complexity define this recruitment pattern. So, and we think there is another physical phenomenon here that is if we increase the slope and the rugosity of the place, the larvae get retained, while if we found this very flat area, just the larvae just get washed away. So there is a higher density because of the complexity of the system, and this slope thing is also important. There are other authors that have found similar patterns that we have found here. So at the same time, in the Canary Islands, we found this alternate stable state. We have seen in the other presentation that this is also occurred in other parts of the world. And at the same time, we have places like El Hierro that has a very high cover of upright seaweeds, while the percentage or the abundance of urchins is very low, while there are other islands that the densities of urchins are very high, and there is almost no algae weeds growing. So, and these two alternate states occur at the same time. But also, we have detected over the years that in the eastern island, where the colder waters are, we have detected there are some sites that were covered by algae, and during the 90s, the urchins just arrived and depleted all the system, all the algae cover. And also, very recently, we have a paper published where we have seen this shift in the state of the system from a barren to an algae system because of the human exclusion experiments, like the marine protected areas. So in just 4 years is the first time that this kind of patterns occur in a short time, in a very short time. In New Zealand or in Tasmania, as Scott was talking about, the recovery requires like many years, like 20 or 30 years, and this has been previously published. So there's a very important species here, and Harris was talking about this earlier in the first talk, during the first talk. So how old are these barrens, or the species? How old are they? How long have been around here? So we know that the Professor Ole Theodor Mortensen from the Zoological Museum of Denmark was collecting diadema from Gran Canary Island around the 1930s. So the urchins were here in the 1930s, of course. But also there is a very nice paper by Johnston at the preceding International Seaweed Symposium, where he noticed that there was vast population of echinoderms were recorded in Lanzarote Island. And this is an actual picture of that year. He said that it's Centrostephanus longispinus, but obviously it's not. It was diadema and forming barrens. Okay, so it was long ago that the barrens occurred. So for sure, the urchins are here this urchin is here since the 30s. And from 1965 to 2012, this urchin is forming barrens, okay? And the barrens are here is a stable state that longs about 47 years. That's for sure because it's already published. But what happened farther back? Harris talked about this before. So these urchins, Harris split the original species Diademantiarum in two populations. The A, that it's the Caribbean, Brazilian, and also Florida species, and Diademantiarum B, that is the population we have in the Eastern Atlantic. It's a new species. So it had been here for almost 2 million years ago. Okay, so it's been here. And evidence of that is that it evolved in a new species that we are going to call, it's now resubmitted for publication, and it's going to be called Diadema africana. It have very different morphologies, like in the pedicellarias and the thickness of the spines. So it's very different from the Caribbean one. So it's a new species. So it's been here for a long, long time. And Harris also talked about these. If we look at the mismatch distribution of the haplotype, the expansion of these urchins occurred once, and it was dated 80,000, between 80,000 and 125,000 years ago. So the urchin was here. What was happening then in the Canaries? The Canaries were warmer. Studying the fossils in Fuerteventura Island, the authors, other researchers, Carracedo and collaborators, have found that this is what they call isotopic period 5E, and it was a warming environment where Siderastrea and Strombus around. It was just when the diadema expansion occurred. So probably the Canaries were like this, with Siderastrea all around, but also diadema. So diadema was here before the humans. So to have a better scale, a human scale, we can take a look at this graph here. What was happening back then with the human, with the hominids? Well, the Homo florensis was in Indonesia, or not, who knows? There's still a discussion about these hominids. And the Homo neanderthalis is similar to my granddad. And we were in Africa. It was still, so we were planning to go out, but we were in Africa at that moment. So such a long time before the humans reached the island, the urchins were here. And I think they were in high densities also. But there are some natural events that we have been seeing over the years. So these long monitorings are very important, as everyone here has been pointed out. So here I'm showing the this is the temperature increasing trend, as everyone have shown before. And in the Canary Islands, the post-settlement survival, this is a model that we have built using the temperature records and also the post-settlement survival of the larvae. And we see here that there are pulses, okay? But these pulses have been more frequent since the 80s, as in the 90s. So, okay, there is such an increase of these densities because of these pulses. But what happened in the field? I'm sorry. So we did this study in uncommon years, so very warm years, where the summer temperature were about 24. And here it used to be very low, like about 23. But now the summer seawater temperature is about 24. And the normal years were very colder. And we found that the settlement was higher during these warmer years. And so there is a correlation between the temperature and the post-settlement survival of these larvae. This is a positive trend, but there are some sites that do not follow this trend. And these sites are in El Hierro. In El Hierro Island. So we decided to study the whole system, the ecosystem entirely. So, so here, what I'm showing is what happened after that settlement, that we have detected in those warmer years. What happened is the densities increase in part of the archipelago, in Tenerife, but also in La Graciosa, MPA. So inside the marine protected area, the urchins increase. So there is some interesting patterns here. But in El Hierro, nothing happens, or even there was a slow decrease of densities. So we decided to study the whole ecosystem. And Sabrina started her PhD work studying the predators of these urchins. But before we go to that, I would like to explain that this system here in temperate areas, there are three main levels that are the producers, the algae beds, the consumers, that here in the Canaries are the urchins, and the predators, the fishes, or the lobsters in other places. And there is a positive control of the algae, but also there is some bottom-up controls of the system. So we wanted to go deeper in these relationships and see what was the forces, bottom-up or top-down. So Sabrina detected a very efficient predators like these ballistids or these labrid that are able to predate upon diadema in very high rates, actually. And when she studied this using tethering experiments through the whole archipelago, we had found very interesting patterns here. So in the eastern island, okay, there is predation. So the community or the fish assemblages that we found at these more temperate areas is not capable of control the urchins. But there are other areas, like in El Hierro and some part of La Palma, that the number of predators is such a huge mass of predators that are able to control the urchins. So there is like a two-part of the archipelago, okay, that the functioning of these parts are very different. Very recently, we have found that, as I told you before, that the exclusion of humans from areas in La Palma, the MPA protection, have served to revert from a barren to an algae system. But there, of course, there is a spatial variation. But in just 4 years, there was an increase of seaweed cover inside the no-take and the reserve area, but not outside. And another example, a natural event, is the underwater volcanic eruption in El Hierro that have very interesting patterns arrived from here. But Sabrina will talk about this in El Hierro Island, okay? So I'm going to just skip this. Another important event that we have detected here is a mortality of urchins. Very recently, we found that this bulge urchin disease occurred in diadema. And this is very rare. The people that study diadema, it's very rare to see this test because they just break down very easily and dissolve it. But so this means that there was a huge mortality here. The mortality started in November in 2009, and then we found out, as other researchers, that the mortality, we found evidence of mortality in February 2010 in Tenerife and in Gran Canaria and in La Palma. So what happened with the population? So the population, in general, there was a spatial variation, of course. There were sites like the 60 or 80% of the population was reduced, but other sites, nothing happened. So there was a spatial variation in this disease event. But the population, as an average, declined by 50%. So it was a mortality event. It wasn't massive as in the Caribbean, but it was a very interesting process to study. So we did some tests with these Petri dishes to find the pathogens that was affecting or infecting these urchins. And we have find out that there is a Vibrio alginolyticus involved in this in disease, and this explosion of the disease. But also there is a neoparamoeba brachyphylla, very similar to the paramoeba that is in the Halifax in the Western Atlantic, Northwestern Atlantic. So there are similar processes here that are occurring in the Canaries and the other part of the Atlantic. So we did some experiments using different concentration of bacteria. We have here Katy and José Carlos and Sabri. And so we found out that injecting the bacteria in different concentrations, overall, there was 100% of disease symptom, of the evolving of disease symptom, but when using the higher concentration. And when we use a lower concentration, there was some urchins that evolved the disease, but others not. So there is a concentration effect here, and I'm sure there is a temperature effect that we have not studied yet. We have seen before neoparamoeba can act as a host for bacteria, so we don't know yet if the amoeba took the bacteria inside the urchin. So we have to keep on doing some experiments. We already inject the Vibrio, but we have to do some injection with amoeba and a combination of the amoeba and the Vibrio, but also to run some kind of experiment similar to Bob, to just put the disease in the water and see if the urchins get infected. But it seems that it is a very similar process. These are the temperature records for winter, for the period where the disease was detected. And what happened in Madeira back then? Well, this is what happened. You know, the temperature was above 22.5. It was the highest temperature ever recorded for winter in Madeira. This is a very important thing because these infections typically exhibit a strong temperature dependence. So we think that this temperature, these very high temperatures developed or was very important to this outbreak of the disease. In the Canaries, what happened in the Canaries? Well, pretty much the same. There are also another period here in the 1998, but the duration of the period was longer here. This is just the average, but here there are like 3 or 4 months with this temperature above 20 degrees. So the temperature is involved in this disease event. That's what we think. And so winters about 20 may promote urchin disease. And what was the effect of population renewal? Because this disease occurred just before the urchins were ready to spawn, and the population declined by 50%. So we wanted to, as in other part of the world, we wanted to know what was the effect on the settlement or the recruitment on the population. The mortality occurred there in the Canary Islands, and as I already told you, the maximum winter temperature was registered there, so about 20. But the settlement, we put these collectors out to see what was, this is as we have seen before, this is the settlement period for diadema. There is no difference with the other previous settlement period. So there was an important mortality event, but didn't have any consequence in the settlement of the species. So the variants are still around and at those places. So the predictions, well, the frequency of mortality and massive settlements will increase in the future, and I will probably aid to the alternation between algae beds and barren grounds. But to explain a little bit the ecosystem function, so Scott has explained before, there is a high resilience of this system, a high persistence of this system in the Canary Islands. But we think as the water warming, there's going to be a lower resilience of the systems that can promote massive settlement in these places, or the mortality events will be more frequent. But this, I think, it will occur at places where the predators are not found. Like in places like this, the predators' diversity is very low, like in this very cold environment here. And so protecting systems or creating or marine protected areas in this archipelago, or at least in the western part of the archipelago, is going to be very important to keep the algae beds stable, because it's like a buffer of water warming. The predators are a buffer. So no matter if we have higher settlement, the urchin predator are going to be there just to stop this recruitment. And well, finally, thank you very much for being here. Thank you very much for the students that are helping us during this conference. Without them, this wouldn't be possible. And the story, of course, continues, but the peak is waiting, so we have to run. Thank you very much.
Thank you.